Hello guys, I'm going to talk to you about this genetics simulator that I show you in the class. You're going to use this to do your genetics lab. We're not going to do the lab in the actual lab. You're going to do it using the simulator. So I will show you how to launch the simulator, how to log in, and how to do the experiment. You will need internet connection to do this, and go to cjslab.com, and you will need an Excel spreadsheet, which is this. You'll not have these information. You will only have this setup in your file that I will send you, and these information you will enter as you do your experiment. Now I'm going to show you how to launch your simulator. When you go to the main page, which is this is the home page, you will see this launch CGS link here. You will click on that link, and this is the simulator. Now you can close this window and make this full screen. When you get the simulator full screen, you click on login to your account. I already set accounts for you using a demo version of this simulator. So since you are a new student, like you're doing the lab for the first time, you click on this new student and collect new organisms. You click on this, it'll ask you the course ID and student account. You will type in demo for the course ID and student account. If you're in T2, you will type in advancement 1. If you're in T3, you will type in advancement 2. Okay, click on this, and it'll ask you if you're an individual or a group. Everybody will do it individually, so you will click on individual and enter your first and last name. After you enter your first and last name, it'll give you a password in case you stop working on your experiment and come back and want to continue where you left off. So I will write like and click next. It has opened me an account, and this you see here, a primer 4981 is my password. I'll write it down. Primer 4981. I don't know if it's case sensitive, so you just try. And I will click on yes, I wrote down my password, and it'll take me to the main page of the experiment. This experiment involves 3 populations. First population has just 1 trait. Second population is also just 1 trait. Third population is studying these 2 traits simultaneously. So this one has eye color trait. Population number 2 has eye shape trait. Population 3 studies both of these traits and tries to understand if they are linked or they're independent. In the population 1, you try to identify the mode of inheritance of this trait. So you want to understand if the eye color is inherited in a autosomal dominant or recessive manner, or it's like incomplete dominance, or it is codominance, or like it is sex linked, or whatever. You try to understand the mode of inheritance in that. And the second one is also the same thing. It is just a trait is different. Trait is eye shape, and you're trying to understand if this trait is inherited as autosomal recessive or dominant, or it is like sex linked, or it's incomplete dominance, whatever. In the third population, you study these two together to understand if the traits are independent of each other or they're linked. So I will not do this. I will not start crossing because I have already done some crossing in another account. I will show you that account. So I will log out. Now I'm going to open the account that I have done some of the crosses of the lab. I'll click on returning student. This is where you go if you stop working on your experiment for some reason and come back to continue where you left off. You click on returning student and enter your password. Remember, the password it has given you was primer 4981, but for me it is different. It is different for everybody actually. It was rain 6962. Okay, there you go. Now these are populations. As you can see, I have already performed like 12 crosses in this, 11 crosses in this, and 3 crosses in this. So for the first population, I'll select first population and click start crossing, and it'll open up this. This is my lab bench, and these are my vials that I have done the crosses. This is the wild population that I have, and each of you will be given a wild population of flies with like certain phenotypes of the given trait. So the trait is eye color here, and I have 2 different type of eyes in the organisms that are sepia-eyed flies and wide-eyed flies. I'll open the data table for this lab, which is eye color. I have eye shape, and I have eye color and eye shape simultaneously. So eye color. And I'll go back to my lab bench. So remember the Mendel experiments. What have Mendel done? The Mendel did, he identified the true breeding individuals first. What true breeding is, whoever you cross that true breeding fly, like you breed that fly or organism to an organism that is similar to itself, they're going to give you the same they're going to give you a progeny with the similar characteristics. Like if you cross 2 white flies, they continue giving you the white flies. So if you cross 2 sepia-eyed flies, they continue giving you the sepia-eyed flies. So what I have done is I just randomly selected flies to cross each other. In the first vial, I have tried. These are the, by the way, each individual is numbered in the simulator. So this fly, let's say, is a fly number 1. You can see right down here. It's the fly number 1, and its sex is a female, and eye color is sepia. You can sort these not like the organisms as labels too. When you click eye color here and eye color again here, you're gonna see them like these guys are labeled as SE, indicating they're sepia-eyed, and these guys are WH, indicating they're wide-eyed. So this individual is number 1 female, and it's sepia-eyed. So what I have done is I crossed. So I did the whites first. I crossed this male parent, which is number 14, to female parent number 60. So fly number 60, which one is this? Is fly number 14, and it's the male fly. And then I moved down until I see a female, because I can't cross this like 2 males, right? So I get this male, this is male, this is male, and this is female. I cross this and this. And how do I do crosses? This is the crossing vial. So I click on that, and the simulator brings it up to this vial, and I click on number 60, and it is also in the crossing vial. Now when I click cross flies, it'll make a cross between these two, and it will give you a resulting progeny in a pop-up window like this, and I'll label it with a file number, whatever. So these are all the progeny you will have, and every trial, every cross will give you 300 flies in the resulting progeny. That's what I set. So I'll clear this. So the cross that I have done first time was this number 14 and this number 60, and I entered it in my data sheet. So the phenotypes that I'm crossing are all whites, and the resulting progeny is in the file number 1. And what cross did I do was I crossed number 14, fly number 14, to fly number 60. And I have done this to see if the flies, if these two fly are true breeding. And if you look at the results of this cross, all whites. I have 300 flies in the progeny, and all of them are white. If you want to see them, you can select eye color and eye color here, and they're all labeled whites. So that means the progeny of this cross 14 and 60 gives you only whites. So that indicates these are possible true breeding flies. I can't say it is certainly that they are true breeding, because white can be a dominant character, and one of these may be a homozygous, which is true breeding. The other one can be heterozygous. I can't say they're both true breeding for now. Okay, I have to do further trials. So I keep this 14 the same, and I cross it to another female. This is 60. 65 is a female, and my second trial was between 14 and 65. So what I do again is I click on 14, I click on 65. They're brought up to this crossing file. Click cross, and it'll give you the progeny. I'm not going to do it because I already done it. Here in the second vial, you have again 300 white flies. If you want to see them labeled here, they're all whites. So that indicates again number 14 and number 65 are possible true breedings. That increases my chance of number 14 is a true breeding fly. So I tried it one more time, like number 14, and the third vial I crossed it to 95. Number 14 to 95, this female, and the result was 300 whites again. They're all whites. So I entered all these informations in the second vial. The phenotypes of the flies that I crossed, they're all whites. The number of the vial is 2, and I crossed number 14 to number 65. The reason I'm doing this cross is to see if the flies are true breeding, and the result, all of them are white, and number 14 is highly likely true breeding. And I have done another trial with number 14 to number 95 to see if the flies are true breeding. In the new progeny, all of them are whites, and number 14 is highly likely true breeding. So after identifying that number 14 is highly likely true breeding, I moved on to the sepia-eyed flies. In the sepia-eyed flies, so starting from number 4, I'm crossing sepia-eyed flies now. And let's see what I have done. I crossed number 1 and number 2 together. If you want to see what these flies are, number 1 is this as a female, and number 2 is this as a male. When you look at this, my chart, always I have written the male first, the male fly, number of the male fly first, and the number of the female fly second, to identify which is which. Where is that? Because like it gives me the male parent first and then the female parent second. So in the fourth vial, I crossed number 1 and number 2 together. They're all sepia-eyed, but in the resulting progeny, 234 of them are sepia-eyed and 66 of them wide-eyed. That means these two parents are not true breeding. They're hybrids, so they can give progeny that is not similar to them. They're sepia-eyed, but they give you a progeny that is wide-eyed. So that means they're not true breeding. These guys are hybrid. So I record this. I use 2 sepia-eyed flies and the vial number 4. I cross number 2 to number 1 to see if the flies are true breeding. These are the results. 234 of them are sepia-eyed, 66 of them wide-eyed, and number 1 and number 2 are not true breeding. This is the result. In the 5th vial, what I have done is I crossed number 3 to number 4. They're all, again, the sepia-eyed flies, and the result is 223 of them are sepia-eyed and 77 of them are wide-eyed. So what I enter this in my document. Again, sepia-eyed flies are crossed. The phenotype is sepia-eyed. Vial number 5, number 3, and number 4 is crossed to see if they're true breeding, and this is the result. My conclusion is number 3 and number 4 are not true breeding flies. In this 6th vial, what I have done, I crossed number 5 to number 9, and the resulting progeny is 214 sepia-eyed and 86 wide-eyed. They again given me wide-eyed flies. That means they're not true breeding. I entered this in my document here. Sepia-eyed flies crossed in number 6. I'm sorry. Number 5 and number 9 is crossed to see if they're true breeding, and number 5 and number 9 are not true breeding flies. In the vial number 7, I crossed number 6 and number 16. How I do my crosses, to just remind you again, number 6, I select number 6. Where is number 6? Here is number 6. By clicking on it, when I click on it, it is brought up to the crossing vial, and move on to number 16. Click on number 16, and it is brought up to the crossing vial, and click cross flies. Why did I select number 6 and number 16? Because number 6 is the male in order. Where is that? Here. And 16 is the female in order. Like, these are all males. And I already used number 9. So after that, male, 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 and 13 is male, 15 is male, and the 16 is the next female that I can use, and I have not crossed before among sepia-eyed flies. So clear this. Number 7, brought up number 7 again, 6 to 16, and if you look at the progeny, they're all sepia-eyed flies. That means either both of them or one of the parent flies are true breeding. So just to make sure, I continue breeding these parents with other flies. So I enter this in my document. Sepia-eyed flies crossed in the vial number 7. Those are number 6 and number 16 flies, to see if the flies are true breeding, and the resulting progeny are all sepia-eyed. They're possible true breeding flies. So I have done another cross in the 8th vial. So here is the 8th vial. I crossed number 6 to number 9, and I told you, remember, number 6 and number 16 in the 7th vial they both can be true breeding, or just one of them can be true breeding. That is enough to give you all sepia-eyed flies. And if you look at this, that number 6 and number 9 is crossed, because I already know number 9 is not true breeding. And when I cross this not true breeding fly to another sepia-eyed fly, and if they give me a progeny with wide-eyed flies, I will understand that 6 is not true breeding at all. So, and that's the case actually. I got 229 sepia-eyed flies and 71 wide-eyed flies. That indicates me that they both have the wide-eyed gene, so they're actually not homozygous sepia-eyed, they are heterozygous flies. So that means 6 is not true breeding. This is my conclusion here. If you can see, sepia-eyed flies are crossed in the vial number 8, 6 and 9 is crossed to see if the flies are true breeding. 229 sepia-eyed and 71 wide-eyed is the result in the progeny. That means number 6 and number 9 are not true breeding flies. Now, to understand if number 16 is a true breeding fly, because I know one of these are true breeding. Now, if number 16 is a true breeding fly, I will cross this to number 5, because I know that this number 5 is not a true breeding fly. So why do I not cross number 16 to number 9? Because they're both females. So I have to cross number 16 to a male, which is number 5 is a male here. So I go to my simulator and show you what I have done in the number 9th vial. I crossed 5 to 16, and I know 5 is not true breeding, and all the resulting progeny is true breeding sepia-eyed. I mean, it's not correct, it's not true breeding. I don't know if they're true breeding, but all the progeny are sepia-eyed flies. That indicates to me that this female parent, number 16, is a true breeding fly in terms of the eye color character. So I identified number 16 and number 14 as true breeding flies. Number 16 is a true breeding fly for sepia-eyed trait, sepia-eyed phenotype, and number 14 is a true breeding male for wide-eyed phenotype. Now I can cross these two together to produce the F1 progeny. Remember, what Mendel have done is Mendel crossed two true breeding different phenotyped plants and get an F1 generation. F1 generation was all resemble to one of the parent. So we're doing the same thing in here. Number 10, I crossed number 14 to number 16. Here is my records. I crossed this time sepia-eyed and wide-eyed flies together in the vial number 10. The flies are number 14 and number 16. The reason I'm doing this is to see if sepia-eyed or wide-eyed phenotype is dominant. And I expect one of the phenotypes will not be observed in the progeny. So when I cross these two together, one of them will be dominant, and I will see whichever that dominant is in the resulting progeny. So if the white is dominant, I will see all whites in the progeny. When you look at the result of the cross, all the flies are sepia-eyed. That means sepia-eye phenotype is dominant phenotype. So I identified that sepia-eyed phenotype is a dominant phenotype. And next, what Mendel have done is he crossed the individuals from F1 together. So I will select one male and one female amongst these. So I'll start from the beginning every time. This is the fly number 2801 is a female, and this is fly 2802 is a male. I crossed these two. They're both sepia-eyed flies, and in theory we know that these guys are heterozygous. So what I predict here, since this is what did you want to learn by doing the cross, and what, if any, assumptions did you make? I crossed two sepia-eyed flies together in the vial number 12, and these are the numbers of the flies. Number 2802 crossed to 2801. I expect to see 3-to-1 ratio between the number of sepia-eyed and number of white-eyed in the F2 generation. So let's see if this is the case. And I did this cross in the 12th vial. I will show you what I have done in the 11th vial. So number 12. Here is the resulting progeny of F2 generation. 225 of them are sepia-eyed and 75 of them are white-eyed. This is a simulator. It's supposed to give me random numbers. It has given me a random number, but I was really lucky that I got exactly a 3-to-1 ratio here. So when I got the 3-to-1 ratio as I expected. So what I can do is I can make a chi-square test to verify my hypothesis, like to decide if I reject my hypothesis or fail to reject that hypothesis. So this is how you do it, do the simulation for monohybrid cross in one trait, that is eye color. You do the same thing for eye shape, and I'm going to introduce you the third population right now, which is the investigating eye color and eye shape traits together to see if they are independent of each other or they are linked genes. So to do that, you go to the CGS again. Go to CGS again, hit save and exit. It'll bring you to the main menu, and you go to population 3. I'm gonna do the population 2 because it's just do the same thing. You investigate just one character and try to identify the mode of inheritance. And for population 3, you click start crossing, and this is your wild population. In the wild population, since you have two different traits, in these two different traits you have 4 different phenotypes. It can be sepia, this is the eye color, this is the eye shape. Eye shape can be bar or lobe eye shape, and eye color can be sepia or white. So eye can be sepia and bar type, it can be sepia and lobe type, it can be white and bar type, and white and lobe type. As I told you before, I have already done all these populations, and I already know which of these phenotypes are dominant. I will tell you that sepia dominant over white phenotype, and bar eye type is dominant over lobe eye type. So since I already know that sepia phenotype is dominant and bar phenotype is dominant, I can try to identify parents that are true breeding dominant for both traits, which means the sepia and bar-eyed, and identify parents that are true breeding recessive for both traits, which are white and lobe-eyed. So I will look for individuals that are true breeding among these guys and among these guys. Since I already know, I'm not going to do any cross to identify the white and lobed ones, because they're recessive for both traits. There's no other way. They cannot be heterozygous. If they were heterozygous, they would have been dominant flies, sepia and bar-eyed, but they are white and lobe-eyed. So I know these are true breeding. I will not do any cross to identify if these are true breeding flies. But I'll do some to identify if these sepia and barite flies are true breeding. So I'm going to show you what crosses I have done. This is the first vial. I crossed parent number 11 to number 7. Number 11 is a true breeding wide and lobed-eyed. Why did I choose to cross these flies to these flies is if these guys are heterozygous, when I cross them to recessive ones, wide and low-eyed flies, I will see some wide and low-eyed flies in the resulting progeny. Actually, I would see half of them to be wide and low-eyed at least. So if this is the case, that means the fly that I have chosen from here is not true breeding. If I cross a sepia and barite fly with this guy, one of these guys, and all the progeny that happen to become a sepia and barite flies, that means that individual I picked from here is a true breeding fly, because it only gives sepia and barite alleles to the next generation, and it makes the next generation dominant for both of these characters, sepia and barite. So what I have done, I did the same thing as I did before. I select the first one in order. Since this number 11 is a male, I selected the female from here. So the first female is this number 7. So I crossed number 1 by clicking on it, and it drags it to the crossing vial, and click on the number 7, and I crossed these flies, and it has given me this progeny. And this progeny, as you can see, has many different organisms. Like it has sepia and barite, sepia and lobide, wide and barite, wide and lobide. That means that this parent number 7, which is a parent with sepia and barite, is not a true breeding fly. And I did not cross, but I put this in my records, of course. Here I have 3 tabs. In the eye color and eye shape tab, I have indicated the first cross that I have done. What I have done is the phenotypes that are sepia and barite flies crossed the wide and lobide flies in the first vial, and the individuals were number 11 crossed to number 7, male and female. To see if the flies are true breeding, I already know that 11 is true breeding, but the results indicated me that number 7 is not true breeding. So I go back to the simulator, and I select number 2, and I show you what crosses I have done. I crossed 11 again to the next female in order. So this is the female number 7, that's a male, that's a male, that's a male, that's a male, that's female, and it's number 26. So the fly number 26, which is a female, is crossed to number 11 here, which is male, and this is the progeny you got. As you can see here, 300 flies, that is all the flies, are sepia and barite. You can see that. I click on all traits and eye color, they're all sepia-eyed, and eye shape, they're all bar shape. Okay. So all the progeny is sepia and barite, indicating that this female parent is a true breeding dominant organism for both traits. So what I'm going to do is the same thing as Mendel did. Mendel crossed 2 parents that are true breeding for both traits. One is dominant for both traits, the other one is dominant for, recessive for both traits. So that means I would cross 11 to 26 for the F1 generation. This is the parental generation. This is F0, number 11 and number 26. they're crossed and they give F1 generation. F1 generation, I already have it in the while number 2, right? So what I'm gonna do is I would self the F1 generation, but it'll give me a like 9:3 to 3:1 ratio if the genes are independent of each other. This is very hard to do. To make my life easier and your life easier, I would cross one of these flies to the recessive parent. Recessive parent is a male, so I will select a female from here. The first individual here is a female and it's a heterozygous individual. When I cross this heterozygous individual to a to this true breeding recessive parent, it will give me 4 different variations with 1:1 ratio. I would give the equal number of sepia barrite flies, sepia lobite flies, white barrite flies, and white lobite flies if the genes are independent of each other. So I give I enter this expectation in my table here. So in the second cross, I crossed sepia and barrite to white and lobite. In the second vial, the individuals were number 11 and number 26 to see if the flies are true breeding, and all the progeny is sepia and barrite, which means they're all dominant for both traits, and number 26 is identified as a true breeding, and this is also the F1 generation. And I maybe expected to cross the F1 generation among themselves. I already told you I didn't do it. To make my life easier, I crossed one of the individual in the F1 generation with this male recessive parent. So again, the phenotypes, the F1 generation phenotype is sepia and barrite, and number 11 is white and lobite in the third vial. And the expectation, if the trait, I said my hypothesis is the traits are independent. So this is my hypothesis, my prediction. If my hypothesis is correct, I expect the resulting progeny will exhibit all possible combinations of phenotypes with the same ratio. All possible combinations of phenotypes are sepia and barrite, sepia and lobite, white and barrite, and white and lobite. I expect them to appear with the equal number of individuals. That makes 1:1 ratio. But the results, as you can see here, I have crossed in the third vial, I crossed this number 11 to number 401, and what I got 138 sepia and barrite individuals. I got 14 sepia and lobite individuals. I had 13 white and barrite individuals, and I got 135 white and lobite individuals. And I entered this in my data table. What we say is these guys, sepia and barrite and white and lobite, are parental generations, like they resemble to their parents. These sepia and lobite and white and barrite individuals do not exist among the parents. They don't resemble their parents. So we call these that are indicated in red, we call these as recombinants. So, and since I don't see the 1:1 ratio, it is very, very different than 1:1 ratio. I conclude that these genes are linked. You can test this using chi-square test. I'm not going to do it in here. I will do it in another video. But what I'm going to show you here to calculate the recombination frequency. So recombination frequency, is equal to number of recombinants divided by number of all the individuals in the new progeny. So, and then in this generation, I know I have like 300 flies, but the number of recombinants are 14 plus 13, which makes 27. 27 divided by 300, and that gives me 9% recombination frequency. Since the starting populations, the wild populations that you are given in the beginning of the experiment, will differ than mine. Everybody is highly likely to calculate a different recombination frequency, so don't care about my the number that I have calculated here, but this is how you do it. So you're expected to do the experiment for all the populations. Let me show you. Save and exit. You're going to do population number 1 first, population number 2 and number 3, and enter everything in the data table like this. Okay. I try to explain everything clearly. It takes a lot of time, I think, but it will help. I think it will help you if you watch the video really carefully, and I hope it is helpful.